Okay, this is the seventh lecture. We're going to finish up biology today, and we got a lot to go through, so hang on to your hats. We're going to go through evolution of the first organisms, primordial soup, Miller-Urey experiment, famous experiments in every biology textbook. We'll go through abiogenesis, evolutionary creation of DNA and RNA, statistical improbability of DNA and RNA, and we're going to go through information theory, DNA and its translation package, irreducible complexity, and then disputed examples of evolution. So there's a lot to go through. This was a pretty bad quote from Richard Dawkins. Richard Dawkins is a world-famous biologist who teaches, I think, biology at Oxford University. And he wrote this book, The God Delusion. He's a devout atheist. And he said this: "We can make the point that however improbable the naturalistic origin of life might be, we know it happened on Earth because we are here." That is so profound. I almost wish they taught logic at even the high school level and the college level. But this is a logical fallacy called affirming the antecedent. It kind of goes, if evolution is true, we would be here. Since we're here, evolution's true. It's a logical fallacy, and it's kind of like saying, well, since we have Christmas sweaters, they must be made by little elves of the North Pole. So same sort of logical fallacy. Let's talk about first living cells. We'll talk more about affirming the antecedent later. It's done all over the place. There's a $17 million evolving planet permanent exhibit at the Field Museum in Chicago. Okay, they spent $17 million on this thing, and they say evolution is one of science's best supported theories. All available evidence, which includes fossils, comparative anatomy, DNA, supports the theory of evolution. Okay, this is blatant brainwashing. It's not even true. Okay, they spent $17 million. They wasted probably some of our tax money. Anyway, 4.5 billion years ago, the Earth was hot and molten, the way the story goes. And the planet cooled and a rocky surface was created. And it rained on the rocks for billions of years and formed a primordial soup. Yummy. And this primordial soup was a bubbling broth of complex chemicals called amino acids. There are 20 different amino acids. And from this bubbling broth of complex chemicals, the first living cells formed. And they call this abiogenesis. A is not, bio is biological, genesis is origin, not of biological origin. It's from a Greek word. And so that's the way the story goes. That's what evolutionists tell you all the time. I don't know if you guys remember from maybe 6th, 7th, or 8th grade biology, but there was something that was believed in the 1800s called spontaneous generation. Okay, they used to think that raw meat that was left out in the sun would spontaneously generate flies just from within the meat. And what they didn't understand is that flies would come, lay their eggs, and then new flies would be produced. Okay? Well, that was really what was happening. Abiogenesis is exactly like spontaneous generation. Okay? Conceptually, they're exactly the same. Okay, Ernst Haeckel said this. He was the same guy we talked about last week with recapitulation, where he forged those embryonic diagrams. He said, Haeckel claimed also that spontaneous generation must be true, not because it had been proven in the laboratory, but because otherwise it would be necessary to believe in a creator. He's biased. Louis Pasteur did real science, and he came up with one of the most important scientific discoveries of our age, which was the pasteurization method, which we use on milk, to canned goods, to almost everything, and it sort of revolutionized the food industry. He basically was saying living things must come from other living things, and if you take a can, you raise the temperature and you kill all the organisms in the can, then the food won't spoil. It'll last a lot longer. Okay, so he basically was saying something in direct opposition to spontaneous generation. Miller-Urey was trying to reproduce the early environment in the primordial soup, the environment that produced the primordial soup. And Miller was a 23-year-old graduate student, I think at the University of Michigan, and Urey was his professor, and tried to scientifically create the primordial soup. And this is a famous experiment, and it has a lot of clout. That's why I talk about it. But he's basically said, Miller-Urey experiment is now recognized as the single most significant step in convincing many scientists that life is likely to be abundant in the cosmos. Okay, so it's amazingly important one, and every science textbook will mention this experiment. Okay, they don't give you the full story, though. I'm going to give you the full story here. What happened is they have this chamber with methane, ammonia, water vapor, and hydrogen. They've got a flask that has a burner under it. It produces water vapor, and you get a circulation around in this apparatus here. And you have two tungsten bits that basically you've got 60,000 volts that go between them, and it arcs. And that's supposed to mimic the lightning in the early primordial world. Okay, so they assume that methane, ammonia, and hydrogen were there in the early atmosphere. Well, today evolutionists believe that the early atmosphere basically came from volcanoes. They say there was a lot of volcanic activity that occurred, and so the gases that are released by volcanoes would have made up the early atmosphere. Well, what kind of gases are released by volcanoes? Okay. It's carbon dioxide and a few other things, but it's not methane, ammonia, or hydrogen. That's for sure. Carbon monoxide, carbon dioxide, and nitrogen gas. Well, carbon monoxide is a toxin. It's deadly to life. Carbon dioxide reduces the ability to produce amino acids, bonds with them, and nitrogen's pretty much really, really stable. It's a monotonic gas. A Science Odyssey, 100 years of discovery, said this: the accepted picture of the Earth's early atmosphere has changed. It was probably oxygen-rich with some nitrogen, a less reactive mixture than Miller's. Or it might have been composed largely of carbon dioxide, which would greatly deter the development of organic compounds. The reason to say nitrogen and carbon dioxide is these are the things that are produced from volcanic activity. Proceedings of the National Academy of Science says, what is the evidence for a primitive methane ammonia atmosphere on Earth? The answer is that there is no evidence for it, but much against it. There wasn't methane or ammonia. And so the next thing is he also excluded oxygen. Okay, if you put oxygen in there, besides from getting a big bang in your apparatus, the whole thing would explode. You won't get any amino acids forming at all. Oxygen's going to bind with them, and you're not going to get anything. But there's a lot of evidence to suggest that ever since creation, there's been an oxygen-rich environment on Earth. Evolution, A Theory in Crisis, written by Michael Denton, this is an intelligent design book, said the Earth has always had oxygen, even more than today. Oxygen is found in the lowest rocks. The only trend in the recent literature is the suggestion of far more oxygen in the early atmosphere than anyone imagined. In general, we find no evidence in the sedimentary distribution of carbon, sulfur, uranium, or iron that an oxygen-free atmosphere has existed at any time during the past span of geological history recorded in well-preserved sedimentary rocks. So they keep looking at these rocks, they keep analyzing them, and they keep finding oxygen in them, and it's baffling them. Here's a high school biology book printed in 2000. And notice before 4 billion years, they have rocks absorbing oxygen. Well, on the vertical axis, it's percentage of oxygen in the atmosphere. And you have it at 0 before 4 billion years. How are the rocks going to absorb oxygen if there's no oxygen in the atmosphere? I don't know. I think it's a mistake. The next problem they had is they filtered out the product. There's no real mechanism to do this in the primordial soup. This is what they say. On a primitive Earth, any amino acids formed in the atmosphere would be destroyed long before they could be destroyed by UV radiation. Even the oceans would not protect them because UV penetrates several meters of liquid water. You can even sunburn underwater. But UV radiation basically destroys amino acids. You're not going to get an accumulation. They claim in most science books, is that goo that Miller and Urey found at the bottom of the flask is rich in amino acids. Okay, well, let's look at that. What was the goo in the bottom of the flask? Well, it was 2% amino acids. Okay, there's 20 total amino acids, and the ones that Miller-Urey produced were mainly of 2 types. Okay, and they didn't have the ability to detect it, but they had very, very small amounts of a 3rd type of amino acid. And virtually all proteins require all 20 amino acids to produce, so you need all 20 in the primordial soup. Okay, I don't consider that rich in amino acids. And then 85% was tar and 13% was carboxylic acid, which are both toxic to life. So you got 2% amino acids, mainly of 2 types, and you got 98% death. That is not a successful experiment showing how life can originate. Okay, beyond that, even if you did have a rich combination of all 20 amino acids, let's assume that you did, it takes hundreds of amino acids to combine to make one protein. Hundreds of these things have to line up in a chain. But they unbond in water faster than they bond together. And so Brownian motion drives the molecules apart from each other. Water is a very, very good solvent. Okay, it dissolves salt, it dissolves sugar, it dissolves a lot of stuff. Huber and Wachthauser used pure solutions of amino acids and tried to get them to join up in the presence of pyrite and iron sulfide minerals. So they're trying to rig a type system where the amino acids will line up in long chains, is what they're doing. They managed to get a few to join up in pairs and a very small portion to join up in threes, but all of these combinations were rapidly hydrolyzed again. Okay, that's a far cry from hundreds that's necessary for even a single simple protein. So the best experimental efforts to prove chemical evolution do just the opposite. They demonstrate beyond doubt that biologically useful molecules do not form spontaneously in water, even in unrealistically pure solutions. So to summarize with the Miller-Urey experiment, there was no methane or ammonia in the atmosphere, so you can't make proteins because of that. There's evidence of oxygen in the early atmosphere, so you can't make proteins because of that. There's no way to concentrate the amino acids, so you can't make proteins because of that. UV rays destroys proteins and destroys the amino acids as well, so you can't make proteins because of that. And the amino acids unbond in water faster than they bond, so you can't make proteins because of that. Okay, there's a whole lot of reasons why this experiment does not show evidence for evolution or abiogenesis. Next up, how many of you guys have heard of SETI, Search for Extraterrestrial Intelligence? Yeah, it's pretty famous. I'm sure you've seen the History Channel shows, Searching for UFOs. It's interesting because what they're looking for is some code or pattern that doesn't exist in nature. Well, if they turned their telescopes into microscopes and looked at the human cell, we've got a code or pattern called DNA that doesn't exist naturally in non-living matter. It doesn't just spontaneously generate. If they use the same criteria, they would see that there was an intelligent source that created us. Any language system is coded information. It always takes greater information to produce information, and ultimately information is the result of intelligence. Isaiah 45:18 says, "For thus saith the Lord that created the heavens, God himself that formed the earth and made it. He hath established it, he created it not in vain. He formed it to be inhabited. I am the Lord, and there is none else." God formed everything on the earth. He created everything, everyone. It's amazing. Evolutionists will look at our human body. They'll see all of our muscular system, skeletal system, nervous, endocrine, and circulatory system, and they're all woven together. And you see that. You see the analogy in the Old Testament of He knit us. Paul says, He knit me together in my mother's womb, talking about we're fearfully and wonderfully made. And that knitting together, the different systems being woven together in such a way that they all work really well with each other, is a really good example of how we were made. But evolutionists will look at this. They will say that we're a product of a stochastic trial and error process in the evolution of our genetic base. But the same evolutionists can walk by a riverbed and can distinguish an arrowhead from the rest of the rocks. Something as simple as an arrowhead, and they can see, oh, that was made by an intelligent source. It doesn't make sense to me. I think they should be able to distinguish both as the work of an intelligent source. DNA stands for deoxyribonucleic acid, and it's basically the programming language that God coded us in. Computers work in base 2. DNA is base 4, basically. DNA was discovered in the 1960s. The scanning electron microscope was invented in the 1930s. And they were able to see a double helix. It's basically like a ladder that's been twisted. The DNA is not the information, but it contains the information for everything that comprises us: our hair, our skin, our eyes, all of our internal organs, everything. And it's important to understand that before we understood about the DNA, evolutionists didn't understand sort of at the base level what must be going on if evolution's true. And this quote says, "It must not be forgotten that mutation is the ultimate source of all genetic variation found in natural populations and the only new material available for natural selection to work on." And so mutation is the basic source for evolutionists of new information. Many will be puzzled about the statement that practically all known mutant genes are harmful. For mutations are a necessary part of the process of evolution, how can a good effect, evolution to higher forms of life, result from mutations, practically all of which are harmful? If you're passing on 1,000 bad mutations for every one, quote, good mutation, as if there was one, then you're never going to get an uphill trend. You're always going to have a 1,000-to-1 ratio of badness going on. Okay, and Paul said the entire creation has been frustrated, not by any will of its own, but by the will of the one who frustrated it. When Crick and Watson first saw DNA through their scanning electron microscope, they said, aha, there's a naturalistic explanation for this. There is no God. And so they see DNA as evidence for evolution. Indeed, both Crick and Watson have been outspoken in their belief that the discovery of DNA structures helped overturn belief in the God of the Bible. Francis Crick has repeatedly said that he sees DNA as a confirmation of evolution, which discredits the God hypothesis. Okay, let's see if that's true. This is from my high school biology textbook, my infamous textbook. It says, "The almost universal nature of the genetic code is strong evidence for evolution of all living organisms from a common ancestral life form." And another way to say this would be, if evolution is true, DNA should be universal in all living organisms. Since DNA is universal in all living organisms, evolution is true. That is another example of confirming the antecedent. And it's a known logical fallacy, and it's just like what I said. Christmas sweaters are all made by elves in the North Pole. Okay, it's funny that they have to resort to those kind of half-truths to try to support their theory. If they had any good evidence, I'm sure they'd quote it. But that's what they're reduced to. Those soporific, sophomoric words, gradually and step by step, repeated incessantly, are aimed at covering an ignorance which is both vast and surprising. And this quote was in 1969, and it's still 40 years later almost. Ignorance about the inner workings of evolution and how it relates to DNA is vast and surprising. DNA is actually evidence for the Creator. Let's talk a little bit about DNA. It's made of 4 nucleotides, adenine, guanine, cytosine, and thymine. And when DNA is copied, it's copied into something called RNA. And the only difference between DNA and RNA is RNA has uracil and DNA has thymine. Okay? And of course, it has a backbone of phosphate and sugars. Okay? If you're going to have something pop out of primordial soup, you've got 2 options. You've either got DNA or you've got RNA. All right? 2 options. Okay, there's a pretty big problem for evolutionists with DNA popping out of primordial soup. Scientists can make adenine, guanine, cytosine, and uracil using rare materials such as hydrogen cyanide and cyanocetylene, as if those would be in the primordial soup. But thymine is completely unsynthesizable without the infrastructure of a living cell. If you can't do it in a laboratory, you can't produce DNA in muddy water. This is not a valid option. And the problem with RNA is that it's extremely fragile. Okay, DNA is what's in the nucleus, and it's not fragile at all. It's a pretty tough molecule. And God used DNA to sort of store our information because it is kind of tough. Well, RNA is not nearly as tough. It's going to break apart fairly easily. It's now generally accepted that RNA is a frail polymer unlikely to have undergone prebiotic synthesis and accumulation. And that was from the president of the National Society for the Study of the Origins of Life. And he would know. A translation RNA is too fragile to have been formed by itself from various compounds floating around in the prebiotic soup. So if university-trained scientists working multi-million-dollar equipped-stock laboratories cannot make DNA and RNA, how can a random action of sand and dirty water produce it in the beginning? How? You can't do it. You can't do it in a laboratory. You can't do it in muddy water. So RNA is not valid. It's fragile. DNA is not valid. You can't produce thymine. And I seriously doubt cyanocetylene was in the primordial soup. So you're all out of piggies. You don't have any more options. What is DNA anyways, though? It's kind of like Morse code. You've got a code here, and if you know Morse code, you can translate it, and you can translate it into help. And if you string it out on a rope, you can get them attached to each other, and that's very similar to DNA. But it's important to remember that DNA is a dead molecule. It can't copy itself. If you had an entire strand of DNA for a single-celled critter in the primordial soup, popping out of the primordial soup, you would not have a cell forming around that DNA. You just wouldn't have it, okay? There's nothing to translate the DNA. Remember how I said the DNA is not the information? The information is coded inside the DNA? Well, you've got to have some sort of translator, some sort of translation package to translate the DNA. It's kind of like having a book of German. Unless you know German, that book contains no information for you. Yeah, it contains information for someone who knows German, but if you don't know German, it doesn't matter. So, okay, DNA is a dead molecule. It can't copy itself. If you need the machinery of a living cell to make copies of a DNA molecule. It may seem as if DNA is the information in your body. Not so. The DNA is simply the carrier of the message, the medium on which the message is written. Werner Gitt, he's a German gentleman, and he has a real thick German accent. Rich and I my good friend Rich is up there too. We got to see him and meet him at the Creation Science Mega Geek Conference. It was a lot of fun. He's an information scientist, and he came up with a way of defining information. Okay, and this may blow some of y'all's minds. I hadn't thought about it until I saw his presentation. But he's come up with sort of 5 levels of defining what information is. It's really fascinating research. He's come up with statistics, how many words or letters are there, syntax, structural characteristics of how information is presented, semantics, from the Greek word semantikos, the proposition, message, or meaning, pragmatics, information invites an action, and then apobetics, since the sender is pursuing a goal, what is that goal? So he's got sort of 5 levels of defining information here. And he's got 10 laws of nature of information. Okay, physical laws are always found by making observations in the physical world. Okay, the law of gravity, you drop something again and again, and you can measure its acceleration, you can measure how long it takes to hit the ground, you can drop it at different heights and do different experiments, and that's how Newton calculated the laws of gravity. That's how he formulated them. Okay, well, Werner Gitt has basically done observation on information, and he's formulated 10 laws of information. A real similar deal. Okay, so the 10 laws of nature of information, L&I. Number 1, a pure material entity cannot generate a non-material entity. Okay, number 2, information is a non-material fundamental entity. Number 3, information is a foundation for technological and biological systems. Every biological system contains information, okay, DNA. Number 6, there can be no new information without an intelligent, purposeful sender. Number 7, any given chain of information can be traced back to an intelligent source. Number 9, information cannot originate in a statistical process. Otherwise, you could do a bunch of statistics on your finances and you could just generate money. That'd be great. So all 10 laws have arisen from observations in our three-dimensional world. So there's 6 conclusions that he's drawn from his 10 laws of nature of information. Number 1, God exists. All life forms contain a code, DNA and RNA, as well as other levels of information. Conclusion number 4, God is spirit. Because information is a non-material fundamental entity, it cannot originate from a material one. We can, therefore, conclude that the sender must have a non-material component, spirit, to his nature. Number 5, no human being without a soul. Refutation of materialism. Because people have the ability to create information, letters and books, this cannot originate from our material portion. Thus, each person must have a non-material component. And number 6, no evolution. The biological information, the foundational component of all life, originates only from an intelligent sender. And all theories of chemical and biological evolution require that information must have originated solely from matter and energy. We conclude that all theories or concepts of chemical and biological evolution are false. The laws of nature, of information, are from observed things. There's no observations yet that have proved them wrong. So these conclusions seem very valid. He's got a whole paper on it. I'm just sort of summarizing it here. So it's really interesting. What is information? Norbert Weimer made the off-sided statement, information is information, neither matter nor energy. Because information is a non-material entity, its origin is likewise not explainable by material processes. So there's even more problems for evolution. Problem number 1, complexity of the DNA system. The code is meaningless unless translated. The modern cell's translation machinery consists of at least 50 micromolecular components, which are themselves encoded in DNA. The code cannot be translated. It is the modern expression of omne vivum ex ovo, everything comes from an egg. When and how did this circle become closed? It is exceedingly difficult to imagine. Chance and necessity. And this was in 1971. They found more micromolecular components that translate DNA later. This is 1984. Any attempt to form a model or theory of the evolution of the genetic code is futile. The code consists of about 70 components, which are themselves the product of the code. I don't think we really know how many molecules are necessary to translate DNA to make it useful. But what they're basically saying here is if you have DNA, it doesn't do anything. You've got to have these micromolecular compounds all in the right quantities in order to translate the DNA to do something useful. However, all of these 70 enzymes and proteins are themselves coded within the DNA. Okay? So you can see evolutionists are at a real problem here because you've got to have both of these in order to do anything with the DNA. Okay? Even the simplest single-cell animals have these. Darwin's Black Box, written by Intelligent Design, Michael Behe, a molecular biologist. He says each of the anatomical steps and structures that Darwin thought were so simple actually involve staggeringly complicated biochemical processes that cannot be papered over with rhetoric. We're going to show you a video, and we're going to look at how a protein is made. And this will help sort of let you guys know how complicated it is. It's not a simple thing. Here's a little single-cell amoeba. Isn't he cute? He's just sort of floating around. There he is. We're going through the cytoplasm now. We're inside his little body. And there's the nucleus. We're going to go through the double lipid bilayer here, which kind of protects the nucleus. There's the DNA. It's housed inside the nucleus for protection. kind of see the double helix there. And there's a specialty enzyme that comes and unzips a very specific portion of the DNA. You need a very specific portion because you want to make a very specific protein. The RNA polymerase comes by and basically makes a copy of the DNA called RNA. And this RNA basically travels outside the nucleus. Here's a gate. It's a specialty molecule that's also coded in the DNA and embedded in the wall of the nucleus, and it allows this molecule to pass through. So here it is, passing through. And you have the ribosome. It's kind of like the factory of the cell. It's where proteins are created. The RNA will join up with the tRNA, and attached to the end of the tRNA are amino acids. And these amino acids join one to another. You basically get long chains of very specific amino acids. This is basically a protein, and proteins make up everything from your skin, your eyes, your muscles. All of the tissues in your body are made of protein. So there's leaving the ribosome. And then there's another specialty organelle, which basically, it's just basically a long chain right now. Okay, and it's got to be folded into a three-dimensional shape to be useful. And so it's folded in a very, very specific way. Your hemoglobin, for example, the thing that carries oxygen in your body, has to be folded in a very specific way or it doesn't function, or doesn't carry oxygen. Okay, so then it's folded. It's almost useful. There we go. It exits. It's been folded. If it wasn't enough, then there's specialty molecules which carry the protein to where it needs to go within the cell. This is a great quote. It seems to sum things up. One must conclude that no valid scientific explanation for the origin of life exists at present. Okay, there's a staggeringly complicated mechanisms that are programmed within your body for life to exist. Second problem. Creating life is statistically impossible as well. Typical DNA of a virus is 5,370 nucleotides. There's 3 million nucleotides in a bacteria and 3 billion nucleotides in the DNA of a typical mammal. Viruses aren't usually considered living by a lot of biologists because they take the machinery of an actual cell to reproduce themselves. They go in and they inject their DNA and basically take over the machinery of the cell to reproduce themselves. So they don't even have mechanisms for reproducing themselves. Let's take the DNA of a virus, for example, 5,370 nucleotides. If you run out the numbers, even if you had everything randomly joining up with each other, it would take 10 to the 600th. A ratio of 1 to 10 to the 600th is the probability of getting it right. Mathematicians agree that any requisite number beyond 10 to the 50th has statistically a zero probability of occurrence. And even that gives it the benefit of the doubt. Just for a comparison, there's 10 to the 80th electrons in the universe. Okay, you basically have to invoke infinite matter and infinite time to say that this is feasible. Okay, cosmologist Sir Fred Hoyle said it was kind of like having a universe full of blind people randomly shuffling Rubik's cubes, all arriving at the answer at the same time. When, for practical purposes, the condition of infinite time and matter have to be invoked in order to make evolution succeed, the concept of probability, the possibility of occurrence, is annulled. If you talk to evolutionists, they'll always say, well, given enough time, even a monkey could type on a typewriter and, write a novel. And do the statistics on it. I don't think it's possible. Problem number 3. 19 of the 20 amino acids come in two forms, left-hand ones and right-hand ones. And when Miller-Urey did their experiment of their amino acids, they got half and half. Okay, they got half left-hand ones and half right-hand ones. Well, living things are almost completely created using left-hand amino acids. Many researchers have attempted to find plausible natural conditions under which left-hand amino acids would preferentially accumulate over the right-hand counterparts, but all such attempts have failed. Until this crucial problem is solved, no one can say that we have found a naturalistic explanation for the origin of life. Instead, the isomer preferences point to biochemical creation. Just makes it less probable. The second problem is when we finally mapped out the human genome, we found that there were less genes than what people originally thought. They originally thought there were 140,000 genes. We found out there were between 31 and 39,000 genes, and some put the estimate at 25,000 genes. And I remember reading an article a few years back when they finished mapping out the human genome that said, oh, look, we're not as complicated as we thought we were. Okay, well, the opposite is the case. We're actually more complicated than what we thought we were. Just for comparison, we've got about twice as much DNA as a fly, and we've got about the same amount of DNA as a corn plant. Just thought that was funny. But what's really going on is you've got one gene that's used to make several proteins. It's staggeringly complicated, but what you've got is different segments of DNA that will be used to make one protein. Okay? And basically, it's a really succinct way of coding information. It's like zipping a file. However, it gradually emerged that most characteristics, even simple ones, are regulated by many genes. For instance, 14 genes affect eye color in Drosophila. Not only that, the mutation which suppresses purple eye enhances hairy wing, for instance. The mechanism is not understood. So what happens when you have a mutation and you've got in one gene, and that gene is used to make 7 other genes? Well, you're going to have 7 effects on the critter. Okay, and that doesn't help evolution. That hurts evolutionists. We're still, a single gene may control several different characteristics. This was particularly bad news with selection, of course. These poor flies being irradiation. In 1966, Henry Harris of London University demonstrated to everyone's surprise that as much as 30% of all characters are polymorphic. That is, each character controlled several different factors instead of merely one. It seemed unbelievable, but his work was soon confirmed by Richard Lewontin and others. Next up, let's talk about irreducible complexity. There's a lot of structures in your bodies, like your eyes, your ears, your digestive system, and even internal structures within cells that are very complex. They take a lot of different components to make them work. And if you remove any one component, they cease to work. Behe wrote a book, Darwin's Black Box. He's a molecular biologist, and he's in the intelligent design movement, but he had a good idea here. He basically said irreducible complexity is kind of like a mousetrap. There's 6 components, including the cheese, and if you remove any one of them, the mousetrap ceases to function. One of the examples we're going to use is the flagellum. The flagellum are those little hairy things that are kind of sticking out of the rear end of that amoeba. They help the amoeba swim. It's got sort of a helical structure to it, and it rotates real fast, and the little critter swims. Annual Review of Biochemistry said, thus the flagellar motor is the output organelle of a remarkable sensory system, the components of which have been honed to perfection by billions of years of evolution. Nice story. And it's really God's amazing tiny motor. It can stop and start. It can change speed. It has a rotational speed of 100,000 RPM. It can move bacteria 15 body lengths per second. And for us, that would be like us swimming through peanut butter because the viscosity when an organism is smaller seems greater. Forward and reverse within a quarter of a turn. 100% efficiency at cruising speed. And uses only 2% of its energy for swimming. And 8 million would fit in the cross-section of a human hair. But wait, there's more. Self-assembly and repair. Proton motive force rotary system. Hardwired signal transduction system with short-term memory. Wow. Okay, so here's the flagellum. It's spinning around there. The little critter's swimming. And it's real similar to a motor. How do evolutionists say that this thing evolved? Well, they use something called co-option. The final evolution of the flagellum might then have involved only novel recombination of sophisticated parts that initially evolved for other purposes. We say all these proteins that compose the flagellum were used somewhere else in the cell, and somehow, through the miracle of evolution, they sort of came together and formed this flagellum that ended up having a beneficial thing for the cell. Okay, that's what they say. Scott Minich, a world-class expert on flagellum, I don't know if he's a Christian or not, said there are 40 different components that are required for a flagellum to function properly. 40. Only 10 components are used in other ways in the cell. If you're going to claim co-option, you should at least have the majority of the proteins used in other places in the cell. Beyond that, you need to have the structure put together right. If you take all the components for a car and you throw them in the garage, you're still not going to be able to drive anywhere. You've got to have them put together just right. Psalm 94:9 says, can the one who shaped the ear not hear? Can the one who formed the eye not see? God formed the complicated structures in our body, okay? For we are fearfully and wonderfully made. A lot of people will talk about irreducible complexity, and they'll try to come up with examples of reducible complexity, where you've got this process that involves 20 different proteins, and you can remove one or two of the proteins, and the thing still functions. And they say, see, it's reducible complexity. And one of the examples they'll give you is they'll say that there's two components in the blood clotting chain. Blood clotting is fairly complicated. There's a lot of chemicals that are involved. And there two are plasminogen and fibrinogen. If the plasminogen is not functioning, the poor little mice suffered from uncleared clots and usually died. However, if there's no plasminogen or fibrinogen that's working, then the little guys lived. That's great. The problem is, it's not irreducible complexity because the blood now ceases to clot at all. It doesn't clot at all. That's not reducible complexity. That is an example of irreducible complexity. And the mousetrap is broken. It doesn't function. Another good example of a complicated structure in our body is ATP synthase. And this is like a little motor that produces the ATP molecule. The ATP molecule is basically the source of power for the cell. Okay, every cell has this, every single one. It produces these little ATP molecules, and these ATP molecules are used to power everything, all of the biological processes in the cell. This spins around at 10,000 RPM, and it produces 3 ATP molecules each time it spins around, so you get 30,000 ATP molecules per second. Now, certain poisons disable this from working, and you die in a matter of seconds. This is a very critical piece of machinery that God programmed into living organisms. So the first organism that had to pop out of the primordial soup would have had to have some power source. It probably would have had to have this somehow popping out of the primordial soup. Here's sort of an example of ATP synthase and the actual molecules, the proteins. Remember how they folded the protein up into its specific shape and how there were mechanisms to actually get it folded right? This is why it's so important. Geometry, I mean, we're three-dimensional structures, obviously, but the geometry is incredibly important. The last thing we're going to talk about are disputed examples of evolution. A lot of people will say, look, evolution. This is proof of evolution. This has a benefit. See, it's a mutation that has a benefit. And they'll talk about sickle cell anemia as one of the famous ones. This is not evolution. And what they claim is that it has a benefit because people don't contract malaria. It is a defect in the blood cell. Okay, and a certain percentage of people who have sickle cell anemia really bad will die with it. It is a genetic defect. It is not a good thing to have this. So the question isn't, is it a benefit? Okay, the question is any new information being added here? Because really what you need to have for evolution to be true is you need to have new information being added to the system. And in this case, you don't have any new information. People will talk about bacterial resistance to antibiotics, and there's a lot of papers written by creationists on this. And I'm just going to go through one example here. There's an ill-fated sailor that died in 1845, and he was found about 150 years later, and he was frozen in ice. It turned out that there was bacteria in its stomach, and this bacteria lived for 150 years. This guy was thawed out, and they took the bacteria from his stomach, and they basically subjected it to modern antibiotics just to see what would happen, to see what 150-year-old bacteria would do with antibiotics. And it turns out that some of them were resistant to some of the antibiotics. It seems like within any population of bacteria, there are some, natural selection, there are some that are going to live. So it's not any new information that's being added here. Okay, and there's a lot of papers on this, and there's a lot of science behind this, and you can get really deep into this, and if you want, you can do that, but we don't have time here. Four-winged flies. Some people say, look, we've radiated these poor flies with radiation, and we produced four-winged ones. Okay, geneticist Ed Lewis showed that 3 strains of laboratory mutant flies could be interbred to produce four-winged flies. The balancers and halters required for flight stability in the third thoracic segment were replaced by 2 new wings. Okay, well, number one, it's unlikely that those 3 specific mutant flies would have interbred with each other in just the right way. Secondly, there's no muscles attached to the second set of wings at all. Okay, so it can't hardly fly, and it's hopelessly non-aerodynamic. And it would never survive or mate in nature, so it would never pass on its genes. And number 5, duplicate organs is not new organs. Okay, what you really need to have are new organs being created, new information being created. This is not new information. This is just a duplication of information that's already there. It's not evolution. It's the corruption of God's creation. How many of you guys have heard of super baby? Really strong children, besides my wife. These little kids have a lot of muscles. They're just really beefy children. A lot of times they're delivered by C-section because they have a lot of muscle. We find the same mutation in cows and mice sometimes. It's basically a mutation in a gene that produces myostatin. Myostatin prohibits muscle growth. If myostatin doesn't work, then your muscles will grow unbounded. And so you get really grotesque-looking cows like that. This is not any new information. It's part of God's creation that has ceased to function. Yes, they're stronger. Yes, that may be in some way a benefit, but this is not an example of new information, which is what evolution requires. Beyond that, these usually would kill both the mother and the calf because you can't deliver them. And they're only delivered now with great trouble and pain. And secondly, they're usually sterile. And a lot of times they die of heart failure because the heart muscle grows too big as well. Myostatin ceasing to function is not evolution. Blind cavefish, not evolution. It has the benefit that they don't have eyes. They're not going to bump their eyes on the side of a cave. And yes, that's a benefit, but no, this is not the creation of anything new. It doesn't matter if it's a benefit. Look for new information. Feather duster mutation. There's some control gene that's supposed to control feather growth. It no longer works. This isn't even a benefit. These animals shouldn't live. I don't know. Featherless chicken, good for KFC, not good for the chicken. Loss of something. So poor little chickens. What can I say? This is not evolution. This is the corruption of God's creation. When someone comes to you and says, this is evidence for evolution, or I've got a good example of evolution, always ask yourself, is this an example of an increase in information or an example of sorting and loss? I think every time you'll see that it is an example of sorting and loss of information. And whether it has a benefit or not, whether you get malaria, whether you don't get malaria, whether you don't want a chicken with feathers on it or whether you do. The mutations in DNA, they don't support evolution. So here's our stool of evolution. Doesn't have any legs. So what's supporting it? Nothing. Blind faith, materialism. I would say stupid blind faith. Okay, at least we have a defendable faith. We can say, yeah, what we believe makes sense. The Bible makes sense. So in summary, abiogenesis is not supported by science. DNA and RNA cannot come from the primordial soup. Proteins can't be created, concentrated, and even if they could, you couldn't form proteins anyways because they dissociate in water faster than they associate. It's statistically impossible, even if you did have the right stuff in there. Formation of DNA comes from intelligence and does not originate from the material world. DNA coupled with information theory screams God. It screams a creator. Irreducible complexity means structures cannot evolve gradually. All we see are sorting and loss of information. Evolution requires, by definition, requires new information. And let me see here. I've got a video here of Richard Dawkins. Richard Dawkins is an evolutionary biologist from Oxford, big name college. He did an interview in 2005 with Answers in Genesis. They were not trying to trick him with this question. But here's the question. I want to make sure you guys understand the question. Can you give an example of a genetic mutation or an evolutionary process which can be seen to increase the information in the genome? Remember, evolution requires new information. And so if evolution is true, can you give an example of any new information that we've ever seen being created in any animal species on Earth? And so this was the question.
Professor Dawkins, can you give an example of a genetic mutation or an evolutionary process which can be seen to increase the information in the genome? There's a popular misunderstanding of evolution which says that fish turned into reptiles and reptiles turned into mammals, and so somehow we ought to be able to look around the world today and look at our ancestors. We ought to be able to see the intermediates between fish and reptiles, or between reptiles and mammals. We ought to be able to see fish kind of on the way to becoming reptiles. But of course that's not the way it is at all. Fish are modern animals. They're just as modern as we are. They're descended from ancestors which we're descended from. Way back 300 million years ago, there would have been an ancestor, which was the ancestor of modern fish and the ancestor of modern humans. And that ancestor, if you could have been there then, you could have seen the first steps towards the fish, say, coming out onto the land and becoming something like an amphibian. But that was a long time ago. You wouldn't expect to see that.
So that was the answer he gave. It cut off the beginning of that clip, unfortunately. But what they actually did is they asked the question, and then he paused for 11 seconds and sat there for 11 seconds, and then he said, please turn the cameras off. And they turned the cameras off, and they gave him 30 minutes to formulate a response. And that was the response he gave. He sidestepped the question. He didn't even answer the question. He said, no, we don't see evolution happening today. That was basically his answer. That concludes biology. Next up, we're going to talk about ancient man and dragons, and it should be a lot of fun.